putting on. Okay. What are we seeing? RNA interference. Okay. So RNA interference, as we are seeing, it is a natural defense mechanism seen in eukaryotes. Okay. So what are we have seen? Viral pathogen, double-stranded RNA. Okay. We will see the processes. I think we only saw the introduction for this RNA interference.
This tissue culture it's not there in old NCERT.
No.
But it's there in social.
Old NCERT it will be there in botany, not in zoology.
Oh.
Okay, so same content will be there in botany in old NCERT.
But which type?
I'll check and I'll tell you.
There was a chart.
Yeah. Okay. So where are we? Yeah. RNA interference. See, normally in a body, what happens is RNA interference. So we will just see the normal defense mechanism first. Okay, so the normal defense mechanism first we are going to see how it occurs in organism. So any eukaryotic cell. Okay. So now let us say your cell only. Okay. So your cell only is going to have its own genetic material. Now a virus is going to send its RNA inside. Okay. So this RNA should be converted into what? A DNA. So what is the procedure here? So the single-stranded RNA is going to turn into a double-stranded RNA, which then turns into a double-stranded DNA, after which it integrates with the host. So this is what happens. Okay. So single-stranded RNA belonging to what? This is belonging to the virus. So this is going to be the viral genetic material. Single-stranded RNA becomes double-stranded RNA, and then it becomes double-stranded DNA, after which only it can integrate with the host. Now your cells, they have developed a defense mechanism. We are talking about what? Eukaryotes. What are eukaryotes? What are eukaryotes? Nucleus, they have a well-defined nucleus. Okay. Yes, cell organelles, well-defined nucleus means everything is enclosed within a membrane, right? So, can you give me examples of organisms that are eukaryotes?
Humans.
Animals, plants, what else? Fungi, except prokaryota. So, keep that in mind. Every animal is going to be eukaryote. Every plant is also eukaryote. So, keep that in mind. Okay, so now this single-stranded RNA is turning into double-stranded RNA. Now, this is the trigger point for your RNA interference. Now, this, when it is detected by an eukaryotic cell, it is a sure way that the cell knows what is present. A virus is present. So, detected by an eukaryotic cell. Something is wrong with the board, but anyway. So, it is detected by the eukaryotic cell. Now, what happens after its detection is that it is going to trigger its own mechanism. This is a very specific set of proteins that are secreted, after which the eukaryotic cell first is going to release enzymes that are called as dicers. So from the name dicer, what do you think happens? What do you think its first function is? It is going to slice it. It's going to cut. Cut what? So this releases dicer enzymes, which is going to fragment the double-stranded RNA into smaller pieces. Okay. So now we know that this is going to be the double-stranded RNA. So which is now ready to turn into DNA through what process? RNA to DNA, reverse transcription. Okay, so this is going to be a whole length of the double-stranded RNA, which is now going to be fragmented by the dicer enzymes. And these fragments are not, don't think that just because it is fragmented, it is now destroyed. No, Here it is doing this for a specific work. So now after fragmentation, we will call this each of this fragment as siRNA or also called as the miRNA. Okay, siRNA is going to be the small interfering RNA. miRNA is even simpler, it is the microRNA. Okay, so those are the fragments called as. Okay, so this is going to be the fragments. Okay, so now the fragments, that is the siRNA or miRNA, I'll just write siRNA, is going to bind with another protein complex that the eukaryotic cells are going to release, which is called as RISC complex. Okay, so this RISC complex it is going to bind with. So now this RISC is going to be a protein. Okay, RISC stands for RNA induced silencing complex. I'll write that, please listen now. So this is going to be the RISC complex, which is a complex protein. Okay, so this is a protein structure. Now each of these RISC complex will pick one piece of this siRNA. So siRNA is going to be picked up or it is going to attach to the RISC complex. Now this RISC complex is very intelligent, we can say. Why? Because it reads the sequence of the RNA. It knows in what sequence the nucleotides are arranged here. Is this belonging to your body? No. This belongs to the virus. So how does it know it belongs to the virus? Because this is very clearly a double-stranded RNA. So what this RISC complex will do is, it now has read the protein, it goes around your cell, wherever this exact same sequence is present, it will destroy it immediately. It is going to silence it. Okay, so that is what it does. So this is for manageable portions. Smaller portions are easily manageable. Each of it will be picked up by a RISC complex, or even if one fragment is missing, that is fine, because wherever it finds this RNA, it is going to destroy it. This signal is quickly passed to other cells also. So every single cell now knows which sequence it should destroy. So which means what? All those sequences are going to get destroyed in your body, thus effectively protecting us against viral infections. Okay. So now what is this RISC? What did I say it stands for? RNA induced silencing complex. Because unless this siRNA or miRNA is found, this is not going to work. Okay, they have to bind together to do this work. So RNA induced silencing complex, what it is going to do? It is going to read the RNA sequence, and it is going to trigger the destruction of the exact same sequence all around the cell. So trigger the destruction of sequence. It should be the exact same sequence. So I hope you all understood how it works. Okay. So it triggers the destruction of this double-stranded RNA wherever it is found in our body. Okay. So this is the normal procedure that occurs. Now how are we going to use it against a nematode? We will see. This one did you all understand? Have you all completed? No. Okay, right. From where? Please write. We have both are same. Both are same.
Detection happens after it gets integrated in the host cell or before?
Before, after it gets, see, once it turns into double stranded DNA only, it can integrate with the host DNA. Even before that, all of these processes will occur. Please write it fast. Keep it. We still have water. RNA interference. Transgenic amines. Medicine. Radiation. Test is at 2:30 only, right? Online people, you are all not writing the test today. Complete fast. You all understand? See, this is the normal procedure that occurs in, even in plants it will occur. If there is a virus that is entering and if it is trying to do reverse transcription, exact same thing will occur. Okay, but it should be an RNA virus. Okay, so only then this double stranded RNA will form.
Where does the RNA list come?
The cell itself produces. Your cell produces. The host cell produces it. The dicer, the risk, everything is going to be produced by the host. Who is the host cell? We are the host cell.
But before it binds with the before that itself the fragmentation takes place, right? The destruction. So why do we need further destruction?
That is not destruction. Okay, so that RNA is fragmented means in that cell only it is stopped. In other cells still integration may occur. Okay, so even further infections it has to stop in new cells. So this is an easier way. Now it can go and destroy it anywhere. This can even destroy the single stranded RNA itself. Okay, so why? Because it knows the sequence. Wherever that sequence is present, it can destroy. So even before it becomes a double stranded RNA, it can be destroyed.
And those fragments need not be the same, like they need not have the same sequence, right? So like when different fragments are found, different nucleotide sequences.
Will be there.
So like then how can it like read one and then send messages to everyone?
The multiple risk factors will bind with all of these fragments, right? Okay, so all are going to bind to some risk factor. And then now they have the whole of the gene sequences because reading a large fragment is going to take them a lot of time. But if they break it into smaller manageable fragments and different proteins are reading it, that will be easier. Okay? Right. Yes.
And how is it that we still get impacted?
How is it?
How is it that we still get impacted by that?
That is what, if you are having a mechanism, they will have a mechanism, right? So some mechanism that is preventing from, see how we saw about restriction enzymes. Restriction enzyme prevent bacterial infection by fragmenting it. But then bacteriophage produces ligases, like that they will have defense mechanism. Okay, so now we are going to use this to control the nematode infection in tobacco. How we are going to see. A nematode is an animal which means it also has RNA interference. Plant is also a eukaryote which means it is also going to be having an RNA interference mechanism. Keep that in mind. So now how we are going to make tobacco pest resistant. Specifically which pest? Nematode. You already saw the name of the nematode? Malay dog and incognito?
Yes.
The spelling that is given for that is very pure in your NCERT. That is not the right one, but still, that is what we will follow. Okay, so nematode resistance. How? See, tobacco plant, we are going to try and protect. So, the gene is introduced in the tobacco plant. For what? The gene for both strands of essential mRNA of the nematode. This line is very important for you to understand. Okay, so now we are introducing the gene in the plant only. But gene for what? For producing two strands of an essential mRNA of the nematode. So, yes, sense and antisense both. Okay, so both sense and antisense strand will now be produced where? In the plant only. Okay. So in the plant, I will explain. Right. In the plant itself, there are going to be two strands of RNA produced. But that strands of RNA is for what? It is for nematodes mRNA. An essential mRNA of nematode means this mRNA of nematode is going to produce some very important protein for that nematode, without which the organism will die. Okay. Think of it this way, we are doing this for humans instead of nematode, put humans. So what we are doing now is, we are introducing a gene into a plant. Gene means what? It's a DNA. It can code for what? It can code for RNA. Okay. So this, it turns into RNA. So now we are introducing a gene in such a way that it's going to produce both the strands of the RNA. Okay. So now in the plant, this double-stranded RNA is now produced. That if a human is consuming, what will happen? We are going to see. Now we are introducing a gene in such a way that the mRNA codes for it for some essential protein. Let us say now we are introducing it for mRNA that produces hemoglobin. That is going to form hemoglobin. Now the person is consuming this plant which has a gene which can also produce a complementary strand for that mRNA. Which means what happens? Double strand is going to be entering into your body. Immediately what your body triggers? The RNA interference, because double-stranded RNA in no other way it can enter according to your body, so it thinks it's a viral infection. So what it does is, it breaks this mRNA that you have consumed with two strands, and it is going to read the sequence, and it is going to destroy any mRNA in your body with that sequence. But what is that mRNA coding for? Hemoglobin. So which means what? Your own RNA interference is going to destroy all your mRNA that is now coding for hemoglobin. It doesn't know that it is coding for hemoglobin. All it knows is that it is double-stranded, which means it's viral, which means wherever I see the sequence, I will destroy. That is all your proteins are going to do. Which means what happens to this person? They can no longer produce hemoglobin, even if DNA is still there, even if it turns into mRNA. Immediately what happens, since you have taken up the gene that is now going to have a complementary strand also, it goes and immediately binds. So what happens? No more hemoglobin. Exactly what happens in a nematode. So in this nematode, since it is also a very primitive organism, the proteins that are essential for its parasitism is what is targeted. Okay. So what happens? It consumes this double-stranded mRNA. This RNA interference works, and then it immediately kills the nematode. Which means what? Who is killing it? The plant's RNA interference is not killing it. The nematode's own RNA interference is killing it. Okay, understood? So this is how we are using RNA interference against that nematode. Any questions in this concept? Anushri?
The plant produces both these strands. Then wouldn't it be
That is what I was waiting for someone to ask. The plant itself is producing double-stranded RNA. Won't its RNA interference get triggered? It will. But it won't have any effect on the plant because it's not the plant's mRNA. That sequence won't be found in the plant at all. So it will also have its own RNA interference triggered, but no effect will be seen in the plant. But still, if fragments are ingested, it is still double-stranded, so the nematode is going to be affected. Okay? Yes, online. Who called? Am I audible?
I only got.
Yes, Anushri.
No, I got that earlier.
Okay.
Could you explain this term that you were
I will write, I will write. So gene for both sense and antisense, which very simply means the double-stranded RNA is now going to be produced. Okay. So antisense I was spelling your children. Sorry. Okay. So essential mRNA of nematode means that this is going to code for a protein that helps in survival of the nematode. So now the important thing is it is the plant that produces the double-stranded RNA. Won't it get digested in the nematode's digestive system? It will, but that is how it is absorbed. But this only works for what type of pests? You can have the pests only. This is the bigger part of the lesson is over. Now we will next or small topics only. Completed. Okay, so we have been seeing about gene addition. Did we see Hirudin? Did we see Hirudin production? No. So we have seen BT and this RNA interference, golden rice.
Golden rays we saw.
Golden rice we saw there. So the next fourth example for gene addition would be Hirudin production in plants. What is Hirudin? What is Hirudin? Where is it produced? Okay, hirudin is a chemical which is produced by hirudinaria. What are hirudinaria? Leeches. Yes. So hirudin is a very potent anticoagulant. Which is now going to be introduced in a plant. This gene introduced into a plant, the plant which is called as Brassica lapis. Know about this plant? What type of plant, at least any idea? Okay, it belongs to wild mustard family. Okay, so this is commonly called as a rapeseed. Have you heard of it? Okay, so this is going to be the plant. So from seeds, hirudin is extracted. Nothing much about this. So this is again adding a gene, a leech gene in these plants, so that it is going to produce a specific type of protein called hirudin. What could it be used for? If people are more prone to coagulation or blood clotting, it would cause these thrombus in their major arteries, can cause a stroke. So for them it can be used to prevent clots. Can it dissolve clots? That is not possible. Okay, so dissolving of clots is not possible. So next is gene deletion. See then the type of plant that is called as the flavor saver tomato. So here we are going to increase the shelf life of tomato. So for that, what we should prevent? By preventing over ripening of the fruit. So how they are going to achieve that is by removing a gene. The gene which is responsible for producing an enzyme, that enzyme which will cause ripening. So removing the gene which is polygalacturonase gene. So what this polygalacturonase sounds like? Ending with A, so it is an enzyme. So this polygalacturonase is an enzyme which will break down pectin. Why pectin? What is pectin? It's a part of cell wall. That is what is making these fruits softer and riper. Okay. So enzyme that breaks pectin, which leads to ripening. So if I remove this gene, will the fruit stop its ripening? Slows it down because enzymes are all catalysts only which can speed up the process. So if it is not there, ripening will still occur but much slower. So which automatically increases the shelf life. So enzyme that breaks down pectin. So here then what is happening? Slower ripening, but what it also did was it increased the flavor also. That is why it's called as the flavor saver tomato. Okay, so this is about gene deletion. So in agriculture, these are all going to be the uses. Next we see medicinal. Okay, so next in medicine where biotechnology is used.
So are you going to learn about beauty?
No. So 12 genetically engineered therapeutic drugs are used in India. Around the world it is 30. Okay, so some of them we will see now. Erythropoietin, tissue plasminogen activator, granulocyte colony stimulating factor. I will tell you what these are used for also. The same thing, granulocyte macrophage colony stimulating factor, alpha interferons, okay, gamma interferons, So what is erythropoietin for?
Species production.
Why would that be needed? For treating what? Sorry. Okay. Erythropoietin is for treating anemia because it is used for RBC production. Tissue plasminogen activator. So it is used in cases of treating heart attack. Colony stimulating factor, granulocyte colony stimulating factors. If it is associated with immune system, mostly it is going to be for cancer. Immunotherapy. And these are are also for cancer, okay, but specific type of cancer. So we'll see that. So alpha interferons are for what type of cancer? Leukemia and other cancers. Mainly for leukemia. And gamma interferons are also for cancer, but it is again for arthritis. What type of arthritis? Okay, so done with this. Thank you. Page is over. Done? Rest are right now. Okay, and then interleukins. We know what is interleukin is for. What is interleukin? Interleukin is for immune disorders. And anti-hemophilic factor against, yes, hemophilia. So this is usually factor 8, which is for hemophilia. What is hemophilia? What is it? Clotting factor, growth hormone, follicle stimulating hormone, insulin, growth hormone for, this is the easiest, for what? FSH is for infertility, insulin is for diabetes mellitus. What is one more? Streptokinase. Streptokinase for clot busters.
Number of follicles in our body is not that how we
Number
of follicles in terms of primary follicles.
that is not for that. To people who have this cystic fibrosis and all, these follicles won't develop easily. Okay, so if follicles are not maturing, this is only for maturation. So follicle stimulating hormone will convert this primary to secondary, secondary to tertiary, tertiary to graphene, right? So for that only it is going to be used. Okay, maturation and production of ova. See, usually in this infertility, fertility treatments they are doing, right? So what they will do is at one time we saw multiple ova should be released. How is that happening? The administration of this follicle stimulating hormone only, which means at a time multiple primary follicles will mature. For that only we are using. I completed
From growth hormones or what?
in children itself you can detect it because beyond a certain age if they are not growing it means that they are going to have pituitary dwarfism, so for that it is used. It is usually referred to as somatotrophin but that is growth hormone only. Okay, right. So these are all going to be the medicines that are used. So now we are going to see about one medicine that is produced in a specific way, that is insulin. So insulin is a very essential chemical that needs to be produced, a protein, a hormone that is produced. So now what used to be the sources of insulin?
Slaughtered cow.
Yes,
Slaughtered cow.
yes, slaughtered cattle from their pancreas. Sometimes even horses from their pancreas. But what is the major disadvantage of this is the yes. So it triggered an immune response. It triggered an it triggered an immune response. Okay. So now that is the reason why we had to produce it through biotechnology. But there was a huge what we can say is a hindrance, we can say a problem was there. A problem is that, see, there is going to be a gene, let us say this is a gene that is coding for insulin in your body. But the problem is that this insulin gene, it produced something that is called as proinsulin. So it produced proinsulin. Proinsulin means what? It is in its inactive form. See, insulin is a very pretty protein. So it is going to have these three strands only. Okay. So there is going to be, please listen this part. So there is going to be an A chain and there is going to be a B chain. Two chains are there, A and B chain. Now in the A chain, there is an intra-chain disulfide bond present. And between A and B chains, there are going to be two interchain disulfide bonds that are present. Only two. Okay, so this is our proper human insulin. But the problem is that there is going to be one more chain that is connecting them, which is the C chain. The C peptide, yes, which is not needed. This is going to be the proinsulin. Each of it is going to be a peptide chain. Okay, so the B chain is, what's a peptide? It's going to have multiple amino acids. Yes, it's not exactly protein. Protein should have more than 50 amino acids, but here it is going to be less than that. Combined, there are only 51 amino acids in a human insulin. Okay, so now this is going to be the problem. So the gene for insulin and produce proinsulin in its inactive form only, which means what? If I am going to use a bacteria to produce, if I am introducing my gene of interest, let us say this is going to be the gene for insulin. So I am introducing it. What this bacteria can produce? Proinsulin only. Another drawback is that this proinsulin can be converted into active insulin in a human system only. In a bacteria, it's a very primitive organism. Naturally, it doesn't even need to produce insulin, which means it can't convert proinsulin into an active insulin. This was the major problem. But this problem's solution was found in the year 1983 by a company called Erel. Did you understand the problem? So here, proinsulin only is going to produce. The issue is that it cannot be converted into active insulin outside the body. Or it can be, but it's a very tedious process again. So Eli Lilly, what it did? So what it did is quite remarkable and unique because see this is going to be the structure of humulin, which is just going to have A and B chain. Okay, so which is going to have its own disulfide bonds. So it is just the disulfide bond that is connecting it. So it is nothing that complex. So the A and B chain. So this is called as humulin, which means it is the active form of human insulin. So what Eli Lilly did was it designed genes for, this is important, for production of A and B peptides separately. Hmm?
Produce separately.
And then it will be joined. So A and B peptides individually, which means what? C not even needed. Okay, so it's not even needed. So A and B is going to be produced separately, which means what? So here they are going to design a gene that is going to code for chain A, which is going to be giving only that protein. So what we have done is inside E. coli, we are putting a plasmid. That plasmid is going to have only the gene for A chain. So A chain is produced. Similarly, on the other side, what we are doing? So there is a gene for producing B chain, and that is going to give us only the B chain. So here there is a different E. coli which is going to carry the gene that will produce only the B chain. Eventually, these two can be joined simply by what bonds? Disulfide bonds. So disulfide bonds will give us the proper humulin. That is all. This is what happened in which year? Years are very important. Which year? 1983. Okay. Understood how insulin is produced? Please write as fast as possible.